Welcome back to the deep dive. Today we are making a pretty significant move. If you've been with us for the last few sessions, you know we've been spending a lot of time in the gut. We've been dealing with what I kind of call the squatters, the pathogens that just hang out in the hollow tube of the intestine.
Right, which biologically speaking is technically still outside the body. I mean, it's a tunnel running through us, but the organisms there are luminal. They are sitting on the surface.
Exactly. It's like standing in the hallway of an apartment building. You're inside the building structure, but you aren't actually in anyone's living room yet. But today we are breaking down the door. We are crossing that mucosal barrier. We're talking about the organisms that swim in the blood, invade the deep tissues, and hide inside the most private sanctuaries of the human body: the brain, the heart, the bone marrow.
Yeah, we are tackling Chapter 73 of Murray's Medical Microbiology, Blood and Tissue Protozoa. And I really have to say, this is where the microbiology gets, honestly, it gets diabolical.
Diabolical is definitely the right word. We're talking about malaria, Chagas, sleeping sickness. These aren't just minor nuisances. These are the absolute heavy hitters of global health. I was actually looking at the stats before we started, and malaria alone still kills nearly 500,000 people a year.
And the real tragedy there is that a huge percentage of those are children. But beyond the epidemiology, what makes this chapter so fascinating and terrifying, really, is the biology of survival. Because when you move from the gut to the blood, the rules of engagement change completely.
How so? Because in the gut, you have the microbiome and peristalsis fighting you. But in the blood...
Well, in the blood, you are swimming in a literal sea of immune cells, neutrophils, macrophages, antibodies, complement proteins. Everything is trying to kill you immediately. It is a highly hostile, high-security environment. So to survive here, you have to be a master of disguise. You can't just sit there out in the open.
So if we're building a mental map for today's deep dive for you listening, what's our anchor?
The anchor is the intracellular hideout. Almost every single organism we discuss today survives by finding a host cell, whether that's a red blood cell, a macrophage, a heart muscle cell, and just breaking into it.
Yep.
They use our own cells as a bunker to hide from the immune system.
So it's essentially a home invasion.
Precisely. And the specific type of home they choose entirely determines the disease you get. If they choose a red blood cell, you get anemia. If they choose a macrophage, you get immune suppression. If they choose a heart cell, you get heart failure. It is all about location.
All right, let's start with the king of protozoa, the one that has probably killed more humans in history than any other single cause, Plasmodium, the malaria complex.
Yes, Murray lists 5 species that infect humans now. We used to really only care about 4. That's Falciparum, Vivax, oval, and malaria. But there is a relatively new player on the board, Plasmodium knowlesi, which we definitely need to get into later because it really changes the rules of the game.
Before we parse out all the different species, I want to visualize the life cycle. I think this is where a lot of med students get tripped up because there are so many names for the different stages. Sporozoites, merozoites, trophozoites. It sounds like a sci-fi language.
It really does. Let's simplify it. Think of it as a 2-front war. Front number 1 is the insect vector, the Anopheles mosquito.
And specifically the female mosquito, right?
Yes, always the female. The male mosquito drinks flower nectar. He's a pacifist. The female needs blood to make her eggs. She's a predator. Now, here's a crucial biological distinction for your mental map. The sexual reproduction of the parasite, where a male and female parasite combine genetic material, happens inside the mosquito.
Which technically makes the mosquito the definitive host.
Correct. Humans are just the intermediate host. We are essentially just a vessel for the asexual cloning phase. It's a pretty humbling thought. To the malaria parasite, we are just a disposable incubator for its children.
Wow. So the mosquito bites you. She injects saliva to stop your blood from clotting, and hitching a ride in that saliva are the sporozoites.
Right, think of sporozoites as the landing party. They are motile, they are fast, and they have one singular mission. Get to the liver, they circulate in the blood for maybe 30 minutes, tops. That's it. Then they exit into the liver tissue and invade a hepatocyte.
And this starts what Murray calls the exoerythrocytic cycle. Exo meaning outside the red blood cell.
Right. This is the silent phase. You don't feel sick yet. The parasite is inside your liver cells, replicating like crazy. This process is called schizogony. It's asexual splitting. So one sporozoite goes in and tens of thousands of merozoites come out.
It's like a Trojan horse. Or a boot camp where they are just building a massive army in secret.
Exactly that. And here's the thing. Because it's happening inside the liver cells, your immune system often doesn't even realize it's happening. It's stealth replication. But eventually, the liver cell gets so full of parasites that it simply ruptures. It bursts and releases this massive army of merozoites into the bloodstream.
And this is where the second front opens, the erythrocytic cycle. The war moves to the blood.
Yes. The merozoite finds a red blood cell, attaches to very specific receptors on the surface, and pushes its way in. Once inside, it goes through distinct morphological shapes that you absolutely need to recognize on a blood smear. It starts as a ring form, literally looks like a diamond ring. Then it grows into a trophozoite, which is the feeding stage, eating your hemoglobin. Then it becomes a schizon, the dividing stage. And finally, boom,
The rupture.
the red cell bursts. And here is the absolute key concept for malaria: synchrony.
This is the part that always blew my mind. They don't just pop randomly. They actually coordinate it.
It's chemical coordination. Millions of infected red blood cells burst at roughly the exact same time. This dumps toxins, parasite debris, and heme products into the blood plasma all at once. Your immune system senses this sudden massive flood of foreign material and completely panics. It releases TNF-alpha, interleukin-1, a massive cytokine storm.
And that creates the classic paroxysm.
The malaria attack. First, you get the cold stage. Uncontrollable shivering and rigors for an hour, because your hypothalamus is resetting your body's temperature set point. Then, the hot stage. High fever, vomiting, blinding headache. Then finally the sweating stage. The fever breaks, and you're just drenched in sweat.
And because the parasites take a very specific amount of time to grow and burst again, these fevers happen in strict cycles. 48 hours, 72 hours, it's clockwork.
Exactly. It's a biological clock ticking inside your blood.
Okay, so that's the general machinery of how it works, but the devil is always in the details. Murray makes a huge point about distinguishing the species because the clinical outcomes for the patient are totally different. Let's talk about the killer first. Plasmodium falciparum.
Falciparum is the one you simply cannot miss. If you're a doctor and you miss this diagnosis, the patient dies. Murray calls it malignant tertian malaria.
Why is this one so much deadlier than the others? Is it just naturally more aggressive?
It really comes down to two things: greed and stickiness.
Explain the greed part.
Most Plasmodium species are picky eaters. Plasmodium vivax, for example, only invades reticulocytes. Those are young, baby red blood cells. And those only make up about 1 to 2% of your total blood volume. So vivax can only ever infect 2% of your cells. It hits a natural ceiling.
But falciparum?
Falciparum doesn't care. Young cells, old cells, middle-aged cells, it invades them all. It can easily infect 30, 40, 50% of your red blood cells. The parasitemia, which is the count of parasites in the blood, just goes through the roof.
So it simply overwhelms the host by sheer numbers. It destroys so many red cells that you get massive, profound anemia. And what about the stickiness?
This is the actual mechanism of death. Falciparum modifies the outer surface of the red blood cell. It builds these little protein knobs. Think of them like Velcro hooks.
Well, Murray mentions these PFE MP1 proteins.
Right. These knobs bind to receptors on the endothelial lining of your blood capillaries, specifically molecules like ICAM-1.
So the infected blood cells get stuck to the walls of the blood vessels.
Yes. They sequester. They completely jam up the microvasculature. Imagine a traffic jam in the tiny vessels feeding your brain, your kidneys, your lungs. Blood flow stops. Oxygen delivery stops.
That's terrifying. So cerebral malaria isn't actually the parasite eating the brain tissue. It's the parasite choking off the brain's blood supply.
Exactly. It's essentially an ischemic event caused by parasitic sludge. And this leads to a highly critical diagnostic point for exams and the clinic. Because the mature, sticky forms are stuck to the vessel walls deep inside the organs, you don't actually see them in a standard finger prick peripheral blood smear.
You only see the babies.
Right. In a P. falciparum smear, you mostly just see the early ring forms. And Murray gives us a few huge clues to identify them. First is the applique or accolade position. The ring looks like it's stuck right on the very outer edge of the cell membrane.
Like a headphone sitting on an ear.
Or a signet ring on the very edge of a table. And second is multiple rings. Because there are so many parasites in the blood and they're so greedy, you might see one single red blood cell with 2, 3, or 4 rings crammed inside it. That is a huge red flag for falciparum.
And what about the gametocyte? That's the famous one everyone remembers.
The banana. If you see a crescent or banana-shaped organism on the slide, it's falciparum, period. No other human malaria makes a banana shape.
Okay, so falciparum is sticky, it has a heavy load, it clogs the brain, and it has a banana shape. Got it. Let's pivot to the relapsers. Plasmodium vivax and Plasmodium ovale.
These two are fascinating because they really play the long game. Remember that liver stage we talked about?
The silent incubation boot camp.
For falciparum, that's a one-way street. They go in, multiply, and leave. But for vivax and ovale, some of the sporozoites enter the liver and just stop. They go totally dormant. They become hypnozoites.
Like hypnosis or sleep.
Exactly. They can sleep inside your liver cells for months, sometimes even years. They are biologically suspended.
So theoretically, a soldier returns from overseas, takes their standard malaria pills, feels totally fine. But 2 years later, they're out shoveling snow in Chicago and bam, their fever spikes.
That's the absolute classic scenario. It's a relapse. The hypnozoites woke up. This matters immensely for treatment because chloroquine kills the active blood parasites, but it cannot touch the dormant liver hypnozoites. You have to add a second drug, usually primaquine or tafenoquine, to essentially fumigate the liver.
Now, structurally, how do we tell them apart on a slide? You said Vivax likes young cells.
Right. Reticulocytes are physically larger than mature red cells. So when Vivax is inside, the whole red blood cell looks swollen and pale, and you see these fine pink stippled dots throughout the cytoplasm called Schiffner dots.
I always use the mnemonic, Vivax is vast, big cells. What about oval?
Oval also likes young cells, so they're slightly enlarged too. But the key is the shape. They look, well, oval. And the edges of the red blood cell are often ragged or fimbriated. Murray describes them as having fimbriated edges, kind of like the torn edge of a piece of paper.
There's a really cool evolutionary biology story with Vivax involving the Duffy antigen. I feel like this really helps explain the epidemiology for students.
Oh, this is one of the best examples of host pathogen coevolution out there. To get inside a red blood cell, P. vivax acts like a burglar with a very specific key. That key fits a lock on our cells called the Duffy antigen.
But some populations change the locks.
Exactly. In West Africa, a genetic mutation arose thousands of years ago that basically removed the Duffy antigen from red blood cells. The people who had this mutation were totally immune to Vivax, so they survived and passed on that gene. Today, the vast majority of people of West African descent are Duffy negative.
Which means you almost never see P. vivax in West Africa.
Right. If you have a patient who traveled to Nigeria and has malaria, it's almost certainly not Vivax. It's likely falciparum. But if they traveled to India or South America, Vivax is incredibly common.
Okay, so we have the killer, which is falciparum, and the relapsers, Vivax and oval. Let's hit the old timer. Plasmodium malaria.
P. malaria is the slow poke of the group. It has a 72-hour replication cycle, fever on day 1, then not again until day 4. That's called quartane malaria, and it strongly prefers old red blood cells.
Senescent cells.
Right. And old cells are rigid. They don't stretch easily. So when the parasite grows inside, it gets squished. It forms this very distinct band or bar shape straight across the equator of the cell. You might also see what's called a rosette schizont.
And finally, the new threat, Plasmodium knowlesi. This is the one you call the imposter.
This is a true zoonosis. It's actually a natural parasite of the long-tailed macaque monkey in Southeast Asia. But as we cut down forests for things like palm oil plantations, humans and monkeys are coming into much closer contact.
And the parasite jumped species.
It did. And knowlesi is highly dangerous because it has a 24-hour cycle. It's the fastest replicating malaria out there. The parasite load doubles every single day. Patients can crash very, very quickly.
Why is it called the imposter, though?
Because under the microscope, it plays tricks on you. The early ring forms look exactly like falciparum, but the mature forms look like malaria. It tricks the lab technician. You might look at it and think it's the benign, slow malaria because of those mature forms, but it's actually the hyperfast monkey malaria.
So if you're looking at a case from Southeast Asia, like Malaysia or Borneo, and you see what looks like malaria, but the patient is getting severely sick incredibly fast, think knowlesi.
Precisely. It's a crucial clinical pearl.
All right, let's leave the tropics and come a bit closer to home. Let's say we're summering in Nantucket or Martha's Vineyard. We're moving to Section 2 of Murray, Babesia.
The malaria of the north.
It's super confusing because the clinical symptoms are so similar to malaria. You get the fever, the chills, the broken red blood cells causing hemolytic anemia. But the vector here isn't a mosquito.
No, not at all. It's the Ixodes tick, the deer tick, which happens to be the exact same tick that carries Lyme disease, Borrelia burgdorferi, and Anaplasma.
Which brings up a pretty scary clinical possibility, co-infection.
We see this all the time in endemic areas. A patient comes in with Lyme disease, they have the classic bullseye rash, but they're much sicker than they should be with just Lyme. Their blood count is dropping. You treat them with doxycycline for the Lyme, but their fever just doesn't go away. You have to check for Babesia.
Now, under the microscope, Babesia lives inside red blood cells, just like malaria does. It even forms little rings. How do we tell them apart?
There are two key clues for Murray to anchor on. First is pigment. Malaria parasites eat hemoglobin and poop out a dark brown crystal called hemozoin. You can literally see this pigment inside the parasite. Babesia, on the other hand, does not make pigment, so the infected cells look much cleaner.
And the second clue is the shape.
The famous Maltese cross. Sometimes 4 Babesia merozoites join together in a tight cross or tetrad shape. It's rare to spot, but if you see it on an exam or a slide, it's pathognomonic. Nothing else does that.
There is a very specific group of people who are in grave danger from Babesia. Who are they?
People without a functioning spleen. Splenic patients.
Why the spleen specifically?
Because the spleen is the blood's primary filter. It's the quality control inspector. It spots infected, misshapen red blood cells, and macrophages physically bite them out of circulation. Babesia is usually a mild, sort of flu-like disease in healthy people, precisely because the spleen keeps the parasite numbers low. But if you don't have a spleen, the parasite replicates totally unchecked.
So the parasitemia just skyrockets.
It creates massive hemolysis, severe anemia, and ultimately renal failure. It can be rapidly fatal.
So if you have a patient who had a splenectomy after a car accident years ago, and they tell you they want to go hiking in Connecticut or Rhode Island, tell them to wear DT and tuck their pants into their socks.
Absolutely. It could literally be life-saving advice.
Let's move to section 3. Possibly the most successful parasite in the entire world, Toxoplasma gondii.
Successful is honestly an understatement. It's estimated that roughly 1/3 of the global human population is infected with this protozoan.
1/3. That's billions of people.
And yet, most of us have absolutely no idea. Toxo is the ultimate master of the stealth approach. But to really understand it, we have to look at the cat.
The definitive host.
Yes. Toxoplasma can infect almost any warm-blooded animal on Earth. You, me, a cow, a bird, a mouse. But it can only reproduce sexually in the intestinal tract of a feline.
So the parasite actively wants to get back to the cat.
It does. And it has evolved a literal mind control mechanism to do it. When Toxo infects a rat, it actually changes the rat's brain chemistry. The rat loses its innate fear of cat urine. It might even become sexually attracted to it. So the rat basically offers itself to the cat, gets eaten, and the parasite completes its life cycle.
That is just wild biology. But for humans, we aren't getting eaten by cats. We usually get it from the litter box and this is a big one rare steak.
Yes, 2 main routes for us. 1 is digesting oocysts from cat feces, which usually happens when cleaning the litter box. 2 is ingesting tissue cysts in undercooked meat. If a cow eats grass contaminated with cat poop, the parasite hides inside the cow's muscle tissue.
Yeah.
You eat that muscle rare, you get the parasite.
Once it's inside us, what actually happens?
It enters the macrophages and spreads rapidly throughout the body as tachyzoites, tachy meaning fast. This is the acute phase. But your immune system is usually strong enough to catch up, and it forces the parasite into a dormant state. It forms bradyzoites brady, meaning slow inside cysts, located primarily in your brain and your skeletal muscles.
And there they sit, potentially for the rest of your life.
Just waiting. Murray breaks down the clinical picture into what I call the rule of 3.
Let's go through them.
Scenario 1 is the immunocompetent adult. That's you or me. We get infected, maybe feel a bit flu-like, maybe get a swollen lymph node. It could look a lot like mononucleosis. Then we're fine. We just carry the cyst forever.
Scenario 2 is the real tragedy, congenital toxoplasmosis.
This is exactly why pregnant women are told not to change cat litter boxes. But here is the critical nuance for medical exams. This severe risk only applies if the woman gets infected for the very first time during her pregnancy.
Why is that distinction so important?
Because if she was infected 10 years ago, she already has circulating IgG antibodies. Those antibodies cross the placenta and protect the baby. But if it's a primary brand new infection, she has no immune defense yet. The tachyzoites cross the placenta and directly attack the developing fetus.
And the damage is severe. Murray lists a classic congenital triad you have to memorize.
Yes. Chorioretinitis, it attacks the back of the eye, often causing blindness. Hydrocephalus fluid builds up in the brain, causing macrocephaly. And intracranial calcifications, you can actually see these distinct calcium deposits scattered throughout the brain parenchyma on a CT scan.
And scenario 3.
The immunocompromised host, specifically patients with advanced AIDS. This usually isn't a new infection, it's a jailbreak. The immune system collapses, usually when the CD4 count drops below 100 and those dormant cysts in the brain wake back up.
What does that look like clinically?
Toxoplasmic encephalitis. The patient presents with new-onset seizures, severe headaches, focal neurological signs. And on an MRI, you see these classic ring-enhancing lesions that looks like a brain abscess or a tumor. In an AIDS patient, toxo is the absolute #1 differential for a brain mass.
Okay, moving on to section 4. We're leaving the blood and tissues for a minute and going for a swim. The free-living amoebae.
These are honestly frightening because they aren't obligate parasites. They don't need us to survive. They happily live in warm, fresh water eating bacteria. But if we accidentally get in their way, it's catastrophic.
Let's talk about Naegleria fowleri, the infamous brain-eating amoeba.
The clinical scenario is almost always exactly the same. A healthy young person, summer, it's a hot day, they go swimming in a warm freshwater lake or a poorly chlorinated pond, they dive in or do a cannonball.
And water shoots forcefully up the nose.
That is the critical moment of infection. The water carries the amoeba high up into the nasal cavity, right to the olfactory epithelium. The amoeba senses the nerve fibers and literally follows them, like climbing a ladder, through the cribriform plate, which is the porous bone at the base of the skull, and goes directly into the frontal lobe of the brain.
It physically crawls into the brain.
It does, and it releases powerful enzymes that literally dissolve the brain tissue so it can feed on it. This causes PAM, primary amoebic meningoencephalitis.
And the prognosis for PAM?
Dismal. It is almost 100% fatal. The progression is rapid and fulminant. By the time symptoms appear, which is usually severe headache, altered sense of smell, stiff neck, the tissue destruction is already massive. Murray notes that unfortunately we usually diagnose this post-mortem on autopsy. But if you somehow catch it early, you are looking for motile trophozoites in the cerebrospinal fluid. You won't see cysts in the tissue because it kills too fast for them to form.
Contrast that rapid death with acanthamoeba.
Acanthamoeba is also free-living in water, but it's much more opportunistic. It classically causes keratitis, which is a severe infection of the cornea. This is huge for contact lens wearers.
Specifically, people who make their own homemade cleaning solution, right?
Exactly, or people who swim with their contacts still in, or who use tap water to store them overnight. The amoeba gets trapped between the plastic lens and the eye, and it just starts eating the cornea. The pain is described as excruciating, way, way worse than the eye actually looks on initial exam.
So Naegleria is the rapid brain eater from the lake, and acanthamoeba is the slow eye eater from the dirty contact lens.
Correct. I should note, acanthamoeba can also cause a chronic, slow brain infection called granulomatous emetic encephalitis, or GAE, but that's almost exclusively seen in immunocompromised patients. Naegleria kills the young and perfectly healthy.
Section 5, we're back to insect vectors, Leishmania.
Now we're dealing with the sandfly. It's tiny, it's silent, and it transmits a parasite that absolutely loves macrophages.
There's a distinct morphology switch here that Murray emphasizes heavily for exams.
Yes. In the gut of the sandfly, the parasite has a long tail, a flagellum. It's highly motile. That's called a chromastigo. But once it gets injected into human skin, our macrophages immediately come over to eat it.
Which is usually exactly how we kill invading bugs.
Usually, yes. But Leishmania actually wants to be eaten. Once it's inside the macrophage, it purposefully sheds its tail to save space and becomes a round mastigote. It thrives in the harsh, acidic environment of the phagolysis zone.
And looking at a pathology slide, these mastigotes have a very specific look.
They are tiny ovals. They look like a dot and a dash right next to each other inside the host cell. The dot is the nucleus, and the dash is the kinetoplast, which is a specialized clump of mitochondrial DNA. If you see a macrophage completely stuffed with dot and dash circles, you're looking at Leishmania.
The disease itself has 3 distinct faces, which honestly sounds like a horror movie spectrum.
It really is. Phase 1 is cutaneous leishmaniasis, often called the oriental sore. It's caused by species like Leishmania tropica or mexicana. You get a bite, and over weeks, it turns into a large, crater-like ulcer with thick, raised borders. It looks like a little volcano on the skin. It heals eventually, but leaves a very disfiguring scar. There's also the chiclero ulcer, specifically on the ear cartilage, seen with mexicana.
Phase 2 is mucocutaneous, Leishmania brasiliensis.
This is the stuff of nightmares. It starts as a skin lesion, but years later, it metastasizes to the mucous membranes of the nose and mouth. It's called asplundia. It literally eats away the cartilage. It destroys the nasal septum. It can leave the patient with a gaping, destructive hole in the center of their face.
And phase 3 is visceral, Leishmania donovani.
Kala-azar, or black fever. Here, the parasite doesn't stay in the skin at all. It goes deep. It invades the entire reticuloendothelial system, the liver, the spleen, and the bone marrow.
So the organs just swell up.
Massively. The spleen can become so enlarged it fills the entire left side of the abdomen. That's massive splenomegaly. And because the parasite physically packs the marrow can't make healthy blood cells anymore. You get severe pancytopenia, low red cells, low white cells, low platelets. Without treatment, the mortality rate is nearly 100%.
So Leishmania is really a spectrum depending on the species. Skin ulcers, facial destruction, or systemic organ failure.
But all of it is driven by the exact same dot and dash amastigote, hiding safely inside our own macrophages.
Final section of Murray, the trypanosomes. We have a vast ocean dividing them here. Africa versus the Americas.
Same genus, but totally different diseases and mechanisms. Start in Africa, African trypanosomiasis, or sleeping sickness.
Vector for this one is the tsetse fly.
Which, unlike the sneaky sandfly or the mosquito, has a horribly painful bite. You definitely feel it. Now, the parasite here, Trypanosoma brucei, stays out in the blood plasma. It doesn't hide inside cells at all. It just swims freely between them as a flagellated trypomastigote.
But wait, if it's just out in the open blood, why doesn't the immune system just blast it with antibodies?
Because the parasite is an absolute master of disguise. It uses a mechanism called antigenic variation.
Explain how that works.
The parasite wears a thick protein coat on its surface called VSG, variable surface glycoprotein. The immune system detects it, takes a few days, and makes a specific antibody to kill that exact coat. But just as the parasite population is about to be totally wiped out, a small fraction of them flip a genetic switch and put on a brand new, completely different VSG coat.
It's like changing the license plates on a getaway car mid-chase.
Exactly, or putting on a fake mustache. The old antibodies are now totally useless. The parasite population spikes again. Then the immune system has to learn the new coat, takes new antibodies, and right before it wins, the parasite switches again. This causes the classic undulating waves of fever. And eventually, the parasite gets exhausted playing this game, crosses the blood-brain barrier, and causes the severe neurological sleeping symptoms: confusion, profound lethargy, and eventually a terminal coma.
There are two types of this, right? East and West?
Yes. Trypanosoma brucei is the West African variant. It's slow, it's chronic, it lasts for years. Humans are the primary reservoir. You often see winter bottom sign, which is painfully swollen lymph nodes at the back of the neck. Trypanosoma brucei is the East African variant. It's rapid, acute, and it's carried by wild game animals like antelope. It kills you fast, often from heart failure months before the sleeping sickness even fully sets in.
Now let's cross the Atlantic to South America. American trypanosomiasis, better known as Chagas disease, Trypanosoma cruzi.
Vector here is the reduvid bug, or the kissing bug. It classically bites you right on the face, usually around the lips or eyes, at night while you sleep.
But the bite itself isn't actually what infects you. This is honestly the grossest part of the entire chapter.
It really is. The bug bites you to take a blood meal. As it eats, it gets full and defecates right next to the bite wound. The bug's poop is absolutely full of infective parasites. The bite itself gets very itchy. You scratch it in your sleep, and you unknowingly smear the infected poop directly into the open wound or into your eye.
Disgusting. And devastating. Because unlike the African trypanosome swimming in the blood, these guys hide inside cells. Specifically, they target muscle and nerve cells.
They form intracellular amastigotes, and the damage is highly chronic. We call Chagas the disease of the megas.
Mega heart, megacolon.
Right. Over decades, the parasite slowly destroys the nerves that control muscle tone, specifically the myenteric plexus. Without those nerve signals telling the muscle to contract, the hollow organs just relax and stretch out. The heart dilates and eventually fails. That's dilated cardiomyopathy. The esophagus stretches so much you can't swallow. Megaesophagus. The colon stretches out so much you lose peristalsis and can't poop. Megacolon.
So it's essentially a slow structural failure of the body's entire plumbing system. Well, we have covered a massive amount of ground today, from the synchronized exploding red blood cells of malaria to the brain-crawling amoeba.
We've built the mental map.
Now, let's test it. Rapid recall time. I'm going to give you a clinical scenario. You tell me the bug and the why, making sure to reference the specific Murray section for our listeners to review. Listeners, take a second and pause if you need to think.
Let's do it.
Scenario 1. A 24-year-old backpacker returns from Thailand. He has a fever that spikes every single day. A blood smear shows early rings that look like falciparum, but the older forms look like bands.
That is Plasmodium nullici. The key clues are the geography Southeast Asia and the quotidian or daily fever cycle. The visual mix-up on the smear is the classic trap described in the Murray Plasmodium section.
Scenario 2. An AIDS patient presents with a new onset seizure. His MRI shows multiple ring-enhancing lesions in the brain. He owns a cat.
That's toxoplasmic encephalitis, caused by Toxoplasma gondii. This is reactivation of dormant cysts due to his low cellular immunity. You'll want to review the Murray section on Toxoplasma, specifically in the immunocompromised host.
Scenario 3. A patient in Brazil has severe trouble swallowing. A barium X-ray shows a massively dilated esophagus. He remembers being bitten by a large bug on his face as a child.
Chagas disease, caused by Trypanosoma cruzi. The megaesophagus is caused by the chronic destruction of the nerve plexus by the infection. Refer right to the American Trypanosomiasis section in Murray.
Scenario 4. A man vacationing on Martha's Vineyard presents with fever and severe anemia. He has no spleen due to a past trauma. The peripheral blood smear shows a Maltese cross formation inside the red blood cells.
Babesiosis. The location in the northeast U.S., the lack of a spleen acting as a huge risk factor, and the pathognomonic Maltese cross make the trifecta. This is covered in the Babesia species section.
Scenario 5. A patient presents with a large ulcerated lesion on their arm with raised volcano-like edges after returning from a deployment in the Middle East. A tissue scraping of the ulcer shows macrophages completely filled with small dot and dash organisms.
Cutaneous leishmaniasis. The dot and dash refers to the characteristic amastigo form, which is the nucleus plus the kinetoplast. The geography fits Leishmania tropica. See the Leishmania section for those details.
You absolutely nailed it.
These patterns are really the foundational language of clinical microbiology. Once you see the mechanisms behind them, you really can't unsee them.
Before we wrap up, I want to leave you with a final thought. We talked earlier about Nalisi jumping from monkeys to humans simply because of deforestation. We talked about Babesia and Lyme expanding their territory because of suburban sprawl pushing into deer habitats.
That's a very stark reminder that vector biology is at its core just ecology. We are an active part of these parasitic life cycles, whether we want to admit it or not.
The more we intrude into these environments, the more we essentially volunteer to become the new host. What other animal-only protozoa are just waiting out there for us to cut down the wrong patch of forest?
A very sobering thought to end on.
Thanks for diving deep with us today. Go open Murray chapter 73. Actually look at the pictures of the banana gametophyte. Look at the trypanosome swimming in the blood. Visualizing it locks it in. We'll see you on the next deep dive.